Well, good afternoon, everyone. My name is James Fink. I'm a senior principal scientist at Q State Biosciences in Cambridge, Massachusetts. I'd like to start by thanking the organizers for the opportunity to speak today. And I'm pretty excited to give everyone here an overview of the Q State's therapeutic platform, and then specifically talk about our antisense oligonucleotide therapeutic for targeting UBE3A for Dup15q syndrome. So let me switch to a pointer view so I can walk you through this. So first, a little bit about Q State and what we do. We're a CNS drug discovery company that combines cellular models, all optical high throughput electrophysiology with AI and machine learning computation to derive new and unique insights into biology with the ultimate goal of bringing life-changing therapeutics to patients. And there are some great talks today about ASOs and other RNA-based therapeutics. And today I'll be talking about ASOs in particular, but really this platform is agnostic to the therapeutic approach. So we could do gene therapies, small molecules, and I'll spend a bit at the beginning of the talk walking through each of these steps, but I do want to take a short second here to really emphasize again what it is we're doing. We have advanced scalable cell-based models of human disease. These are primary human tissues, iPS-derived neurons, cell types from other species that are relevant for toxicology and toxicity studies. And through our unique sort of measurement tools and some of our molecular biology tools and engineering in-house, we are able to record neuronal activity in a high throughput way that allows us to get information from around 500,000 cells per day. And you'll hear that a little bit throughout the talk. But the ultimate goal of all that functional data is to sort of deduce functional phenotypes that are disease specific for a particular target. And then using that established phenotype, find drugs or therapeutics that can bring, sort of bridge the gap between the wild type controls or the healthy controls and the disease states. And we can use these tools to understand on-target versus off-target effects. And really the idea is to find the best drugs and the drugs that are most differentiating in terms of rescuing the entire phenotype rather than just one or two of the parameters. And the high throughput ability of the system, I think, really allows us to assess that. So now I'll just walk quickly through each of the individual pieces of this particular platform. We have a lot of experience here at Q State working with hospitals and academic institutions in acquiring patient samples for diseases of interest. So this is through sort of IRB protocols and sort of the transfer of materials from those institutions to Q State. We then reprogram those cells into induced pluripotent stem cells that then can be derived into the cell type of interest depending on what disease we're actually interested in for a particular target. As I mentioned at the beginning, we can also utilize cells from other primary tissues and other species, especially when it comes to some of the downstream development work with toxicology and sort of understanding how these molecules might work in relevant tox species. Of course, we're talking specifically today in this talk about the central nervous system. So for that particular tissue, it's really important the cell type of interest that we're making. And so we've spent a lot of time in the past 5 years optimizing protocols to generate several neuronal cell types from motor neurons, GABAergic neurons, excitatory neurons, and dopamine neurons. And of course, again, depending on the target and disease, we'll sort of determine which of those cell types it is that we decide to work on. The other unique thing here is we have protocols and we really excel at producing really large batches of billions of neurons that allow us across many tens of cell lines to generate enough cells to perform entire sets of phenotyping and drug screening with the same batch of cells. And the point there is that you get really enhanced reproducibility and robustness, which of course is important for drug discovery. You don't want your assay to be noisy, that you have to produce a batch of cells every time. And that's, for those of you who are unfamiliar with this particular type of cellular model, that's a challenge in this field. So alone, those cells are really powerful, and people have used them successfully now for a little over a decade for understanding human disease. They have the mutation of interest. If you get them from patients, So there's sort of disease relevance there. There is some immaturity in terms of their function, and that's also well known, and people have been working on doing that. And even more recently, there are a lot of really interesting 3D models that solve some of the complex three-dimensional challenges that these cells have sort of had a hard time bridging in the past. From a way we approach phenotyping at QState is we do this bidirectional approach. So in parallel, we both screen CRISPR edited induced pluripotent stem cell models, as well as those derived from patient and often familial controls. The idea is to get you to the same answer, and that would really give you the confidence that your phenotype is disease relevant and is of target relevance. This is something that's a little easier said than done. Again, there's line-to-line noise, and so of course you want to be able to do this across tens of cell lines to really establish confidence in your phenotype. And because of the sort of scalability and platform we built at QState, we can do this in a really fast amount of time. And so the illustration here at the center is that using our optical electrophysiology tools, we can establish these electrical fingerprints from a disease state. And for instance, in this case, a wild type state where you see the purple and the orange are very different from each other. And the overall idea here is just an illustration, but that this should look the same between the CRISPR edited mutants versus the actual patients that we're trying to treat. Now, where the, I think, real uniqueness comes is through QState's OptoPatch platform for measuring electrophysiology through light. To do this, we use sort of two tools. There are these molecular tools, these optogenetic proteins that we express in cells with lentivirus or transfection. There's two of them. There's Sheriff, which is a blue light activated channel rhodopsin, and Quasar, which is a voltage reporter protein. And they are sort of activated and read out by separate wavelengths, and that allows us to both stimulate and record voltage in cells simultaneously. To be able to do that, we also have custom in-house built microscopes that allow for the measurements of these particular readouts in a high throughput form. There are two main instruments in-house that we do this with. There's the Firefly. This is a 96 well to 384 well format that measures one well at a time and up to 500,000 neurons per day, as I mentioned earlier. And then we also have a Swarm, which allows parallelized optogenetic recordings. So this does 12 to 24 wells all at once, which allows for 10,000 wells per day for a 384 well format. So you can actually screen many thousands of drugs per day with the Swarm, but the resolution there is a little bit less than something like the Firefly. And I'll talk about that more specifically in a second. Now, the way that we express these proteins into neurons, again, that are either CRISPR edited or derived from patients and controls themselves, are by lentivirus delivery. And we can measure two important outputs of neuron function. So those of you that are not as familiar with neuronal physiology, there are pretty much two main ways that neurons are communicating in the brain, which sort of ultimately ideas would underlie lots of the disease states that we would be interested in. There's action potential firing, so we sort of relate to what's called intrinsic excitability. So this is how the neuron is functioning independent of any sort of input from other neighboring cells. Often this is ion channel related, but there are lots of other proteins we know now that can sort of change and somehow on downstream effects on actual intrinsic excitability and what we call action potential firing. And of course, synaptic activity, which is the small units of neurotransmission between neurons, where there's a presynaptic or a sort of neuron who's talking, and then a neuron who's receiving that input on the other side of the synaptic cleft. And again, this is important for several neurological disorders, including autism spectrum disorders, Alzheimer's disease, and several neuropsychiatric disorders. And we have different ways of measuring both of them. I'll touch briefly on both of them in upcoming slides. But this measurement of actual neuron output through electricity, but viewed in a way of output of this change in fluorescence, really gives us the ability to understand important neuron behavior that we think would sort of be important for understanding disease. So what you're seeing at the top is this movie, and you've seen this now several times. This is an actual field of view from one of our experiments There's something like 300 to 400 neurons shown in this movie, and every flash you see is actually a neuron firing a set of action potentials. And our in-house machine learning and computation platform can pull out each individual neuron and extract then the actual voltage traces, in this case derived from fluorescence changes, for each individual cell in a way that has the same resolution as patch clamp. Patch clamp electrophysiology is sort of the gold standard for how this has been done in the past. You take an electrode, you sort of put that underneath a microscope, and you approach a cell with a micromanipulator, bring it close to a cell of interest and impale the cell very gently, or you can suck up the membrane and poke a little hole in it. And it's a little bit disruptive, but you're able to get really high resolution single cell recordings of neuron function in this way. These are actual Optopatch recordings. And for those of you who've done patch clamp, these look almost identical to the types of signals you'd get from single cell patch clamp. And that's again, because we're able to extract the information from each pixel associated with each neuron. And then with the blue light stimulation and the red light recording, understand how these neurons are behaving. I come from an electrophysiology background, so I can appreciate sort of in particular this technology. So I would like to highlight a little bit about this throughput to give you some perspective. My graduate work, some of which I'll talk about today, took 5 years to patch something like 10,000 neurons. And again, that's because with stem cell models, you have to record from several lines, and ideally both CRISPR edited and patient control. And so it takes a really long time when you're going cell by cell by cell. And many of the phenotypic differences at the cellular level for neurological disease can be pretty subtle. CNS has been really hard to develop therapeutics for that reason. And so by recording, lots and lots of cells at once, we can do something like, again, 500,000 times a day, which is sort of 50 times more cells patched in one day than it took a graduate student 5 years to patch. So we can do a lot in terms of therapeutic screening in a really quick amount of time that otherwise would take a really long time with manual patch clamp. Now, with that data in hand, and I think this has been referred to several times today about sort of this big data approaches, these really are ultimate big data sets for neurons. There are billions of neurons in brain with trillions of synapses. So just thinking about neurological disease, there's a lot of complexity there. And so these types of data sets may be some of the largest neuron functional data sets in the world. And that allows us to mine the data and look for really unique insights into disease. And the goal of the computational part of this platform is to identify the unique differences between disease state, as I mentioned earlier, versus the controls. And we can view that as this multi-parameter readout on the very right, what we call a radar plot, where the control conditions are plotted usually at the center and normalized. And then each of the disease states or disease with rescue is indicated by these different colors. And you can see that there's this, what we'll call a fingerprint in red. So there's this specific shape that has to do with each of these unique features. So for instance, some of these features are moving towards the outside in disease state, whereas other features like this one here are moving towards the inside. And the goal is that when you add a therapy or you're screening a therapy on this fingerprint, that you see these features move back towards the control circle at the center. And so we can get a good understanding of what drugs modulate one feature, which modulate two, and which modulate all of them. And as I said at the beginning, the goal is to find differentiating molecules that can rescue entire phenotypes and not just subsets. There may be also interesting things to learn here about a drug if it rescues one phenotype versus the other, and that allows you maybe to do some target deconvolution. I mentioned synaptic activity, and I'll just talk about this very briefly before I move on to some of the therapeutic development for UB3. Is here, we're able to express those two proteins, the channel rhodopsin and the voltage reporter, in separate cell populations by using Cre. So we deliver Cre at a really low dose. We can tune it to the proportion of cells we're interested in discovering and understanding. And then we use a Cre-on version of these reporters and channel rhodopsins and a Cre-off version that then expresses them in orthogonal, non-overlapping populations as seen in the center here. So you have a green population and a red population. And you can stimulate the green population with blue light and record signal from the cells in red light. For those of you that are used to looking at synaptic activity, say in your own lab or in papers, it's often depicted as these downward deflections, because in patch clamp, you measure these as currents, but we are measuring voltage, so they are going to be positive going. And you can see that these postsynaptic potentials, we call them, which are the signals received by the neurons after neurotransmission, are similar to what you'd expect from traditional patch clamp electrophysiology. And here we're just showing that if you apply blockers of these two glutamate receptors, which are one of the primary receptors of glutamate in the brain, you get a reduction in the signal as expected, showing these really are signals being modulated by these particular targets. In this case, AMPA, which is a glutamate receptor with a faster transmission, so you get this reduction in the entire signal, and NMDA, which sort of has this slower onset, where you see here the drug modulation is more towards this later part of the signal. So most of the early part stays the same, and you're really getting a reduction in this decay component. We're not just tackling functional data here at QState. We realize and think it is really important to understand cells as a whole. And the point is we like to correlate the function with other readouts like proteomics, morphological readouts, synaptic morphology readouts, and number readouts, and things like transcriptomics and metabolomics. And combining those two together can provide really powerful insight into disease. And so we actually have, for several of our therapeutic discovery programs, provided that type of analysis and try and learn something new about the biology when you think about both the, say, transcriptomics changes with the functional changes. So here's an example of the synthesis of this platform. So this is for a small molecule approach for a genetic epilepsy. And this genetic epilepsy has an approved drug for treatment. We've derived the cells of CRISPR-Cas9. We also collected cells from patients, and we established a morphological phenotype. So the cells with the knockout of this gene have much larger soma size, as you can see in this image. And 3 different knockout CRISPR edited lines have a deeper spike shape and a reduced spike width as compared to the average wild type in green. When we look at these features across both CRISPR edited and patient controls, as I mentioned for our approach, we see that separation of knockout versus controls and patient versus controls is almost identical. And more importantly, that difference, as you see here, the pink versus the black, or the pink versus the green in the radar plot, is rescued by the approved drug. So this phenotype that this platform has identified is disease relevant, as it can be rescued by the clinical drug. And then we use that phenotype to do a much larger compound screen of many tens of thousands of molecules, with the idea of finding new molecules for medicinal chemistry. So onto the sort of ASO topic for today, and how can we combine this platform with actual ASO therapeutics for treatment of an important neurological disease. So this group knows that you can give ASOs directly to the CNS without having to worry about the blood-brain permeability differences by intrathecal delivery. You can modulate the gene up and down depending on the type of ASO you're giving. In this case, we'll be talking about overexpression of a gene. And there has been FDA approval for ASOs for several neurological disorders, and more recently there are some in the early phase clinical trials for Angelman syndrome, which is another disorder related to this gene. So this is an overview of the current therapeutic programs we have at QState, both small molecule and ASO. Several of them are epilepsy or neurodevelopmental associated disorders, but we also have several programs in pain. And today I'll talk to you about our development of an ASO for Dup15q syndrome. So Dup15q syndrome is a severe neurological disease caused by duplication of a region of chromosome 15 that houses many tens of genes, but there is one important gene, UB3A, that is also overexpressed. It's characterized by hypotonia, motor delays. Many of these patients present with autism, and there is an increased risk of sudden unexpected death from epilepsy. And our therapeutic approach, given this excessive UB3A in these cells, is to take an ASO gapmer to engage RNase H and reduce the UB3A levels, with the idea of reducing the disease burden on these patients. We've spent now 2 to 3 years developing an in-house bioinformatic pipeline for ASO design with the idea of tiling regions of interest, avoiding unwanted and sort of potentially toxic effects. And we try and do this as best we can in silico. And we're also still sort of refining these by further in vivo studies to really build even better rules as we move forward. So for example of UB3, from sort of a list of thousands of ASO molecules, we only synthesized 139 that are likely to modulate UB3A without interfering with other critical genes, other cellular pathways, and have sort of reduced risks of problematic sequence motifs and other sort of off-target alignment problems. And what we're looking here on the left is the in vitro screening in both control fibroblasts and DUP15Q fibroblasts of these 139 ASOs, where we identify several candidates in the top right that have greater than 80% knockdown of the target. And some of the example qPCR data can be seen here on the right, where we've just shown a subset of these ASOs. You can see many of them are knocking down UB3 almost to nothing as compared to the controls, and even in some cases better than siRNA. We can do this in dose response. Of course, we don't want to knock down UB3 too much. So we're looking also for ASOs who are able to reach sort of desired levels at sort of lower doses to avoid things like toxicity or tolerability issues. So there's some example data here on the left for that. And the right-hand side is also just a subset of ASOs with a heat map of percent knockdown. And you see this nice gradient of red to green, showing that these ASOs are approaching 100% knockdown as you get up to 200 nanomolar. Again, these are all in fibroblasts so far. Of course, the protein is sort of the thing we actually want to modulate in these cells. And so it's important to check that we can do that in vitro. So these are also fibroblasts treated with several ASOs. You can see in the Western blot on the left that we have almost complete loss of UB3 protein with ASO treatment. That's similarly here in the Western blot quantification. And then actually a bunch of our ASO candidates, our lead candidates here showing almost 100% knockdown of UB3 protein with 200 nanomolar treatment on fibroblasts. There are mouse models available, and we actually have licensed this mouse model from Dr. Ben Philpott's lab. And interestingly, these mice have an increased risk of death upon chemically induced seizures, which is reminiscent of the sudden unexpected death from epilepsy seen in patients. And so the next step here, one of the next steps is to screen some of our ASOs in the mouse model to show proof of concept rescue of this disease relevant phenotype. I mentioned at the very beginning of the slide deck that we like to check these in several different species, especially ones that are going to be used for some of our toxicology studies. And here we're showing knockdown of mouse UB3 in primary cortical neurons, in primary rat cortical neurons, and in non-human primate fibroblasts. Finally, this is some of my own graduate work. It's sort of pretty serendipitous that I've been able to still work on DUP15Q. I spent my graduate degree at University of Connecticut understanding the functional phenotypes of DUP15Q syndrome using iPS-derived neurons and established this sort of hyperexcitability phenotype in these cells that's not seen in some other related diseases. And the idea here is to use our platform, which I showed you is sort of really well suited for understanding this neuron function to establish those disease fingerprints and use ASOs to rescue that multiparametric phenotype. And that's something that's ongoing today. We've acquired these cell lines in-house as well and have licensed them from the University of Connecticut. And we're just showing here that the cell lines that are from the DUP15Q patients both have increased protein expression and at the both protein and transcript levels. It's important if we're going to be able to screen these cells for phenotypes that we can actually get our ASOs into these cell types and not just fibroblasts. So here we're showing data from iPS-derived NGN2 neurons treated with several of our ASOs, and we show that we can get something beyond 90% knockdown for some of those ASOs. And then even in human primate cortical neurons with gymnotic delivery, do we get close to 80% knockdown in several of these. Finally, is the last piece of data before I summarize here is We're in the early stages now of putting these molecules in vivo. Of course, that will be the ultimate test for target engagement and tolerability. We've identified several leads to move into this phase. So this is just an initial pilot where we ran a 10-day study of tolerability. So the ASOs are delivered to rats intrathecally on DIV0. They're measured for weight and some behavioral observations for every few hours for the first few days, and then once a day up until day 10. And then they're sacrificed and several tissues are harvested. The cerebral cortex is an area of particular interest for this disease, so that's where we're interested here. You can see that there's no difference between the ASO treated group and the vehicle treated group in terms of their weight gain, and all signs of the animals from a behavioral perspective was they were normal, which is a great sign for our molecule. And then here we're looking at the average vehicle treated animals versus the UBE3 expression in 5 different rats, each treated with the same ASO. And there's some variability here, but the idea is that there are up to 40 to 50% knockdown in several of these animals, and that combination of knockdown with the behavior suggests that this is still well tolerated. Of course, we still need to push the dose, but again, those studies are ongoing. So just to sort of summarize the end here is the next step is, in addition to sort of more in vivo studies, is to build this multi-parametric phenotype using our tools, which again are, I think, uniquely suited for this particular disease and the sort of established phenotypes in the literature for that disease, and use it to find the best ASOs to eventually move in for sort of downstream development and actual clinical trials. So just in summary, we've developed a platform that integrates in vitro cellular models with high throughput electrophysiology and machine learning based analytics to find new therapies and differentiating therapies. We're applying them now to ASOs to really find the best ASOs, not just in terms of their in vitro molecular biology profiles, but their actual functional profiles for disease rescue. In Dup15q, we've found a set of lead candidates with greater than 80% transcript knockdown, and that's also true for protein, and minimal off-target effects. And so far, we've tested some of these in vivo and showed they're well tolerated and engage target in relevant brain regions. So with that, I'll take any questions and sort of happy to also follow up later if there are any others that we can't fit in this session. Thank you for your time.